Okay, well, good afternoon, everybody. I'd like to welcome you all to the soil science seminar. Well, this is the penultimate soil science seminar of our semester. We'll have one more next Wednesday. As we started last Wednesday with the student seminars, we're going to continue the student seminars today. Next Wednesday, I just want to give you a clue. Next Wednesday will be really different. Of course, today is going to be kind of different standards too. You'll find out. But next Wednesday will be quite different. But I just want to leave that as a teaser. So you want to come and find out why it's so different next Wednesday.
You told everyone.
Okay, so today is going to be different. We have two types of presentations. The first one is going to be a 12-minute presentation that Khaleel will be presenting, and that's modeled on a standard presentation of a professional meeting. And then there will be time for your questions following her presentation. Then we will go into a lightning round of 4 presentations. Each one will last about 5 minutes. And I ask you to hold your questions for those 4 speakers until the end of the 4th presentation, and we'll have some for questions for all the speakers at the time. So if you have a piece of paper and a pencil, I'm talking to you folks online too, you might want to use that to write down your questions so that you don't forget what they are in the minutes while we're doing it. Okay, so let's start with Ala's presentation. I'd like to introduce you to Ala Khaleel. Ala grew up in Amman, Jordan. She got a bachelor's degree in water environment at the University of Jordan. She came to the United States, got a master's degree in sustainable agriculture here at Iowa State in 2018, and now she's working on a PhD, and she is jointly advised by Marshall McDaniel, Reid, and she close to being done with her PhD. She's going to talk today. The topic is the effect of cropping systems on soil enzyme measurements and methods.
Okay, great. Is it good? Do you hear me? Okay. Thank you, Dr. Thompson, for the introduction. And as Dr. Thompson mentioned, today I'll be talking to you about one of the projects I have been working on, and that's talking about the effect of cropping system on soil enzyme kinetics and measurements. I would like to start the presentation by just answering the simple question of why would we care about enzymes, or who would care about them? And the simple answer to me is that I think enzymes have a very high potential in our soil science community that we as a community haven't tapped into it yet. In science community, specifically soil science, we're all interested in soil organic matter and the decomposition process. And I think we often give microbes a little bit more credits in the decomposition process. And when in fact, if we think about it, it's the microbes that are mainly produced by these enzymes, it's the enzymes that mainly produced by these microbes or plants and other small soil animals. So I think enzymes have high potential because they do the job of mineralizing organic matter and converting the organic forms into forms that are more available for plants uptake or microbial assimilation. Enzymes also have important role in nutrient cycling, and they can be used as a proxy for microbial activity. Therefore, their potential enzyme activities have been proposed as a soil health indicator by the NRCS and the Soil Health Institution. And when we measure enzyme activity, the assays are widely employed and also powerful. We did this in a very simple lab incubation in which we add a very specific substrate concentration to a soil sample, and the substrate is the organic compound that the enzymes would act on. However, since we're talking about potential here and not actual activities, we have to take care and make sure we're optimizing the assays we're following or the protocols we're using to assay the activities. One of the most important things we have to think about and optimize is the substrate concentration, which is basically how much do I have to add to a soil sample in order for me to assess their maximum potential and make sure that they are saturated with the substrates and work as fast as they could. This is often overlooked. No one does that. It's usually they use one common substrate concentration from the literature. It's a lot of work, but we're going to see why it's worth it at the very end of the presentation. More about enzymes. So we all know that enzymes are proteins. There are catalysts. They speed up the rate of a specific reaction. So when we talk about these catalysts and the rates, we're dealing with kinetics here. And in enzyme kinetics, we're interested in looking at how the rate of the enzyme catalyzed reaction will change as we change the substrate concentration. Now, this type of discussion on enzyme kinetics will show up usually on a graph like this. That's the Michaelis-Menten plot, or the substrate saturation plot, in which it has the substrate concentration on the x-axis and the velocity, or we could say the rate at which the reaction or the enzyme catalyzed reactions proceed on the y-axis. If we look at this graph, or we could determine two important parameters from this plot. The first one is the Vmax, which is the maximum velocity of the reaction or the maximum ability of the enzymes to speed up the reaction. And once we determine the Vmax and divide this Vmax by 2, we can draw a line and then go back to the x-axis. We can determine another important kinetic parameter, which is called the Michaelis constant, which is the Km here, which reflects the amount of substrates that it takes to get to half of the Vmax. So in order to measure their potential, we have to make sure we're adding more substrate constant, we're adding more concentration than this Km. The objective of the one thing I want to mention more about this plot, it's very specific for the concentration of enzymes we have in a soil sample or in the reaction environment. So if we have, for example, different cropping systems that we're interested in, we could expect to see different Vmax and Km value if we know that these cropping systems have, for example, different organic matter, different concentration of enzymes, and so on. So it would be, in principle, if we need to understand more about these kinetics, we could okay, I'm going to jump over to the objective of this study, in which we looked at how the three cropping systems impacted the enzyme kinetics for two enzymes involved in the carbon and nitrogen cycling enzymes. And for the study sites, the soil was collected from COPS experiment site, and we had 3 treatments: maize, soybean, prairie with no fertilizer, and then fertilized prairie. So we wanted to compare how these 3 treatments impact the kinetics. So moving on to the results, I have here the Michaelis-Menten plot for beta-glucosidase, which is one of the enzymes that's responsible for the final step of cellulose decomposition. So it will release glucose. That's an important carbon source for these microbes. I have here the Michaelis-Menten plot for the 3 treatments: the fertilized prairie in green, prairie with no fertilizer in orange, and then maize and corn, or corn soybean, in yellowish goldish color. On the x-axis, we have the substrate concentration, and on the y, we have the V-gaze activity, And from this plot, we can see that the Vmax was significantly higher in the treatment, in the peri-fertilized treatment, as compared to the other treatments. For the KM, it was not affected by the cropping systems, but it was just the Vmax that responded to the cropping system. This is another enzyme that's responsible for the nitrogen cycling, or call it the nitrogen cycling enzyme. And it breaks down the peptides or proteins to release amino acids, the leucine aminopeptidase. And for the same thing, I have the Michaelis-Menten plot for this enzyme. And we saw a higher Vmax under the prairie with no fertilizer treatment as compared to the other two. That's expected because microbes could be limited here with nitrogen, so they produced more of the nitrogen cycling enzyme to acquire more nitrogen. KM was also not affected by cropping system for the leucine aminopeptidase. One last thing I want to mention about the implications of KM and why do we need plot before moving to the conclusion. Now, if you wonder about the vertical dashed line in the previous figures, that's actually the commonly used substrate concentration in the literature. So the 0.04 millimolar per gram of soil was started in 2002 by one paper, and then many other papers used the same concentration without checking for how much substrate do they need to add to their soil. And if you remember from the first slide, when I talked about the lack of optimization and the lack of standardization for the protocols and how they can lead to inaccurate measurements and also inaccurate interpretation of the enzyme activities, especially if we were to compare 3 different systems or different cropping systems. Our results from this study support the idea or the need for determining the substrate concentration and not using the commonly used substrate concentration. For example, here I'm showing the Michaelis-Menten plot for the lipase, in which we found that the Km values ranged from 0.22 to 0.25 millimolars per gram of soil. Now, you could see that this is greater than the 0.04 millimolar per gram of soil that is commonly used in the literature. Another thing I did here, I ran some statistics and we were not able to find or to detect any treatment differences if I were to use the lower substrate concentration. But when using higher substrate concentration, we could still see the treatment or the significant differences between the treatments. This is really important thinking about the future research on enzymes, specifically when we want to detect the differences between multiple treatments and come up with interpretation of what does the data really mean or the results mean. Take-home messages, we saw that for both enzymes, Vmax was, the values for Vmax were significantly higher in the perennial system as compared to the annual cropping system, like the corn-soybean rotation. Km was not affected by cropping systems. However, for both enzymes, our saturation substrate concentration values were higher than the commonly used substrate concentration to assay enzymes, which highlights the need for constructing an plot for maybe not every soil sample if that's too much, although that's the recommended, but maybe for each treatment per study site, or if we know that there are some differences in the soil properties at our study site. With that, I would like to acknowledge Dr. McDaniel's and Dr. Thompson's lab, and I'm happy to take any questions.
Thank you, Alan. Dr. Horton has this one.
Thanks for your presentation. Spend some time telling us about the official. So, I didn't quite catch the source of the enzymes or mechanism by which enzyme system.
Sure. So, yeah. So Dr. Horton is asking about the source of these enzymes and the mechanisms of,
So that there will be different enzyme
yeah, why would be different enzyme activities for in different cropping systems. So the source of enzymes could be microbes and or plants, like fruit exudates or animal, like the small animals in the soil. But a lot of studies have found that mainly they're coming from microbes. And when we expect to see higher enzyme activities with these in these cropping systems, it's mainly related to the microbial community and the activity in that cropping system. So if we have a cropping system with more nutrients or more carbon input or nitrogen, that we expect to see stimulation of these microbial activity and the microbial community, we would expect higher enzyme activity. However, that's the simple answer. Sometimes it's more complex. It depends on the microbes. What do they need to acquire? Producing these enzymes is really costly for microbes. So sometimes it's more of the cost versus the demand of what they really need to acquire from that system. Yeah, okay.
I think earlier in your presentation, you indicated that soil type might be a factor for how much substrate is needed.
Mhm.
What do you have in mind by soil?
Well, I think what I was referring to here, I sampled from a study site that's approximately close in the location. However, if I looked at multiple soil types with different soil properties or different soil series, I included many other soils, I might expect to see some stronger effect on the substrate concentration thing. And I just wanted to highlight that if you are planning to measure the enzyme assays on, let's say, a broad region, this is when it gets complicated and you have to check the substrate concentration and not treating all of your soils the same and adding the same substrate concentration. Because the way I think about this, if you have one soil sample with higher enzyme concentrations versus another soil sample with lower enzymes, it doesn't make sense to add the same concentration of substrate to both of these samples. Because again, we're assaying the potential, it's not actual. So we need to make sure we're capturing their full potential for the enzymes in that soil sample.
Last question?
Follow-up question to that. Is there a downside to this pattern?
Yes.
What is that downside?
There are mechanisms for when you add too much, you could see inhibition of the enzyme activity. And that's actually, there hasn't been a lot of study or haven't been a lot of studies like studying this specifically because soil would have a lot of other things like the clay surfaces, organic matter. So we did not fully understand how this impacts the enzyme substrate complex. However, I see this as another reason of why we really need the Michaelis-Menten curve. Like you could tell from this curve, if you are using a high substrate concentration, then you would expect to see some inhibition as you go with the Vmax. I've seen this in some papers, and that's one of the reasons you need that value of Km, and then before you run your actual assays.
Okay, thank you very much, Alan. Appreciate your thought.
Thank you.
Yep. So we're going to take a minute here to get set up for the lightning round.
This is great.
Thank you very much, students, for helping get this set up. Okay, so there will be 4 talks, each one 5 minutes. The first one is Reid Barker's presentation on phosphorus stratification field.
Thank you, Dr. Thompson. Like I said, today I'm going to be talking a little bit about the general topic of my thesis. So for those of you who were here last week to hear me give my 12-minute talk, I'm going to just articulate a little bit in more detail phosphorus stratification in reduced tillage systems and other BMPs. So just as some general background in phosphorus cycling, in typical situations, this is what you'd expect to see. But in reduced tillage systems and in situations where you're reducing the amount of mixing in your surface soil layers, your inputs are going to start to become more and more important, because mineral phosphorus or mineral pools of phosphorus remain somewhat consistent with time. And in the long term, when you're increasing organic matter inputs or broadcasting surface fertilizers or applying manures that will mineralize, and as I'll mention, phosphorus is generally pretty sticky, so it won't move that much like nitrogen, is over time, when they mineralize without mixing, they might remain on surface soil layers. And so that kind of gets to what stratification of phosphorus is best management practices like no-till and cover crops. They often increase the amount of soil organic matter and reduce tillage. And crop residues, broadcasted fertilizers, and broadcasted manure and other sources of phosphorus nutrients can accumulate on the surface when tillage doesn't occur. You can see here in this diagram, this is a pretty pertinent soil stratification paper from Robbins and Voss, 1991. And looking at this figure, in the top part, their control was a ridge-till plant system, where they're at the very surface, about 80 milligrams per kilogram of phosphorus was what they saw, and then down to 20 milligrams per kilogram is what they saw around 20 centimeters. But then comparing that to no-till, which was in practice for 10 years at the point of the study, they were as high as 160 at the very surface, and then just 10 centimeters later, they were as low as 40. So there's a very dramatic disparity between the surface and the lower surface layers. And that some of the implications of that is there's been some studies that have found that increasing the amount of phosphorus on the surface includes orthophosphate phosphorus forms, which can run off in water and be immediately bioavailable when they're lost in subsurface drainage and surface runoff events. And not necessarily which caused this fish kill in northeastern Iowa, but that was just a picture that I took last summer at a lake that I fished quite a bit that had a really dramatic hypoxic event. Another potential issue is poor plant accessibility in dry conditions. So kind of what I would equate that to is it's in no-till situations, typically moisture is what allows phosphorus to move a little bit more, especially plant available phosphorus. And in no-till situations, usually moisture isn't a problem, but in really, really dry conditions, roots might have a much harder time moving through the soil profile to reach phosphorus that's higher up in the profile. It's kind of like trying to reach snacks on the top, at like the top cupboard without like a step stool. And another potential issue is it can obscure soil tests. So in a highly stratified soil, and you can see in this diagram that I made here, is these green bars kind of show off like potential phosphorus concentrations. And if you take a soil core that's too shallow, then you might overestimate the amount of phosphorus, your soil test phosphorus, than if you get it a little more, take a deeper core sample, and that way you'll have a more representative soil sample. And so what can be done about it is you can either implement more aggressive tillage, kind of move away from, like if you're doing no-till, kind of switch to like maybe conservation till management, like disking, But if you're a diehard no-till guy, then maybe there's a lot of literature that suggests that applying phosphorus as a subsurface application would be the best way to go, so that way plants don't have to reach up high to go grab it. And if you don't want to do either of those things, then you can manage your your surface water outflow or tile drainage outflow with catchment wetlands, riparian buffer strips, infield prairie strips, and grass waterways. That's what I got.
Very much, Reid. Okay, we're going to go on to our next speaker.
Thanks.
Microphone set up. Jessica Nelson is going to talk to us about pore space in and around aggregates that, and the way it influences soil water holding.
Great, thank you, Dr. Thompson. So yep, I'll be talking about soil structure and water holding capacity and some of the research that I have done on this and as part of my PhD dissertation that I'm writing. So to begin with, why do we care about soil water holding capacity so much? And it's important for plant access because without water, plants can't grow and then we can't have food. And then climate change is happening, so there's more need for farm resiliency. So understanding how the water holding capacity changes when there's times of drought and how that also impacts the ability for the soil to catch and hold water during intense rain events. And my overall research question for this specific presentation will be, does the structure and pore space within and between soil particles or between aggregates increase the water holding capacity of the soil? So here's a diagram. Soil structure is really important because you can see that there are these macropores where air and water can flow through the soil. We have mesopores and micropores within the aggregates and between the aggregates that help hold that water there for plants to access it. And so the hypothesis that I have is that when you have samples of soil aggregates that are from miscanthus and maize, which are the two crops treatments I looked at in my study at the LAMP sites across Iowa. So when we looked at the soil and the aggregates from these and we destroyed them, I think that that will have the water holding capacity decrease because, as you can see in this image, when you have intact aggregates, you have some pore space between them. And when you destroy them, it kind of makes it more close together. So you lose some of that pore space, and thus I'm thinking that the water holding capacity will decrease. So the experimental design for this was that we had the intact aggregates, what we're calling them, the ones that haven't been destroyed. And to destroy them, we put them in a mortar and pestle. And then what that did was destroy the aggregates and grind them up. But we tested it to make sure we didn't change the soil texture so that we didn't destroy them to that extent, but that we kept the same soil texture, but still getting rid of that structure within them. To measure water holding capacity, we used the funnel filter paper drainage method, where we were measuring the amount of water that was retained in the soil after we saturated it and drained it for 6 hours. Here's an image of what our samples look like when they were intact, and then when we put them in a mortar and pestle, and what they look like destroyed. The results that we got indicated that for most of the soils, the water holding capacity decreased when the aggregates were destroyed. So you can see on this graph, we have water holding capacity over here on the y-axis, and then on x-axis we have our treatments. And these are the two sites that I looked at in the LAMP site plots. So we have the northwest site and the central site. And the yellow is the maize and the green is miscanthus. And then the solid is the intact aggregates, and the diagonal lines indicate the destroyed aggregate results. And you can see here that for the northwest site, there wasn't really a significant difference between any of them, whereas at the central site, we did see that there was a significant decrease in water holding capacity when we destroyed them versus when they were intact. And we are thinking that this is due to water held by that aggregate pore space. And there was one instance where the water holding capacity seemed to increase in the maize at the northwest site. but that wasn't significantly different. But there could be something happening in that soil where when we ground it up, we might have increased surface area, and so that was having some kind of impact for that specific site in that specific treatment. But this really needs to be done on more sites, on more soil types. These were clay loam soils, and so if we did it on more sites, we could probably see a better trend and get a better idea of how that is actually influenced. So my possible conclusions are that the structure and pore space within and between soil aggregates may increase the water holding capacity of the soil at certain sites, which is where we are right now. Because, you know, it's very small, and we really should look at other ones before we can draw any concrete conclusions. So thank you.
Okay, our third lightning talk is presented by Max Ennis. And this topic is root identification and intensified cropping systems.
Thank you, Dr. Thompson. As Dr. Thompson said, I'm going to be talking. Does that sound? Does it sound weird to you guys?
Yeah.
Let's see. Down, further. Okay. I keep breaking this. Is that? Take two. Oh, that's too much. Okay. Take three. Is that?
It's still too much.
I'm not moving it very much. There we go.
That's good.
Okay. So as Dr. Thompson said, I'm going to be talking about root identification in intensified cropping systems. The main questions I'm going to be answering are what kind of methods are out there and what are the advantages and disadvantages of each method I'll be talking about. Oh. To start us off, I want to talk about a little bit about why I'm interested in root identification. I work on the Regen PGC project, which many of you might know is looking at developing essentially turf grasses to plant cash crops like corn and soybean in between. There's a lot of environmental benefits to this practice. You have less nutrient leaching, you've got carbon sequestration, and there can be potentially yield benefits for both the crops if optimized correctly. However, that optimization requires characterization of competition both above ground and below ground. And so understanding how the roots are interacting and if they're having a negative impact on each other is necessary to direct the development of the perennial ground cover system. However, identification of roots is really difficult. When you're above ground, you can see the plant. It's very clearly a soybean, a corn, plot of grass. Below ground, it's a little bit more difficult because roots can look, very similarly, especially when they're a bit more closely related, like grasses and corns might be. The kind of gold standard in corn intercropping studies is visual identification, but there's a lot of reasons why you might not want to do that, because but it really boils down to it being sort of a judgment call on the researcher. Furthermore, the intertwined growth of roots can make identification really hard, and getting good biomass estimates in small samples nearly impossible. So we've got to think about identification methods for roots. The first one that popped in my head, I think the first week I started my studies here, was using genetic testing on roots. There's a few, it's nice, it's accurate, it can tell you what species you've got there. There's a few difficulties with it though. One, it's very expensive. It's also time consuming to develop the assays needed, especially if you're working with a new cultivar of grass that doesn't have assays present already. And it's susceptible to contamination from previous years. If you're picking up an old soy rotation when you want to be studying corn, you might pick that up in your identification, and that can be useful, but it can also just add noise. The other method that has been widely used before is carbon-13 isotope analysis. Plants all have a different diet of carbon isotopes that they take in and analyzing the roots for their carbon-13 isotopes can give you an estimate of what's the biomass and presence of the roots in your particular sample. Again, it runs into an issue where you might get some noise from roots. It's also a little bit expensive, but it's a good tested method for simple root identification, and you can get some biomass estimates from it. The third method is near-infrared spectroscopy. This is essentially using a fancy camera that takes a really wide, wide spectrum of light in, and it can give you a lot of information on what you're working with. And kind of averaging your two spectral curves can allow you to identify what roots specifically you have here. The limitations of it is it's in agronomy essentially untested for root analysis, and it's rarely used for ecology root testing and food grade root tests. But the benefits of it can be enormous. It's a very cheap method to run once you have calibrated, and it's very quick. It's a massive improvement over visual identification once calibrated, and it might give us greater richness to our root data than the carbon-13 isotopes and the genetic testing can give, because it might give us clues to chemical interactions that are going on in the soil. And ultimately, we're hoping that it grants us a deeper understanding of the role that roots play in intercropping and perennial ground cover systems. Thank you.
Thank you, Max. Really lightning, right? So number 4.
Okay, can you hear me?
Will return.
Is it good?
to-
Oh, okay.
Make sure the mic is on.
Okay, is it better? Okay.
Let's talk again.
Do you hear me?
Yeah.
Okay, so hi again, everyone. Now I'll be talking about a different project, but just briefly. I'll be discussing the different nitrogen rates and 2 cropping systems on the impact of microbial biomass and also enzyme activities. So just an introduction, we all know that nitrogen is important and can positively impact the crop yield, but we also know that excessive nitrogen can have a lot of negative implications for the environment and also can impact soil properties in a variable and inconsistent way. For example, I have a meta-analysis here in which they looked at the nitrogen impact on the microbes and found that as nitrogen increases, there was a decrease in the microbial biomass by about 15.4%. In contrast, I have a recent, actually not that recent, but another meta-analysis in which they looked also at the impact of nitrogen on microbial biomass, but they found an increase of about 15.1% in microbial biomass carbon as nitrogen increases. So now we have 2 different contrasting findings from 2 different studies. As for the impact of nitrogen on enzymes, the study that was published in 2016 looked at how different nitrogen rates would impact enzymes. For the carbon cycling enzymes, they found that the nitrogen rates increase the activity of these carbon aquating or cycling enzymes, whereas the nitrogen cycling enzymes were not affected by the nitrogen rates. However, at their highest nitrogen rate, which was greater than 150 kilograms per nitrogen per hectare per year, they found a suppressive effect of nitrogen on enzymes. So the activity of these nitrogen cycling enzymes went down. So as you can see now, we're not still fully understanding the impact of nitrogen on microbial biomass and also on the enzyme activity. Therefore, our objective in this study is to hopefully better understand and impact the effect of 4 different nitrogen rates and 2 cropping systems on these 2 soil properties. We sampled from one side of the LAMPS experiment. We had 2 cropping systems, the maize and then the miscanthus, 4 different nitrogen rates. The highest was almost like 340 kilograms nitrogen per hectare per year. And we did sample in 2022, that's about 7 years after establishment. So we have here the miscanthus and then the corn. And miscanthus have a very thick layer that's been found to decrease soil temperature, increase soil moisture, and also enhance the labile carbon inputs. So when this carbon goes to the soil, we would expect to see stimulation of the microbial activity and community in the soil under miscanthus. So we would expect to see higher microbial activity in the miscanthus as compared to the corn system. I don't know why it's not showing, but for the nitrogen, we expect to see a decrease in the microbial biomass as nitrogen increases. There should be a background, a dark background here. It's not showing. Anyway, our results, what we found for microbial biomass, we did not find any significant differences. So microbial biomass was not affected by either cropping systems or nitrogen rates. I have here corn in yellow and miscanthus in green. We could see that there was some decline in microbial biomass as nitrogen increases. However, again, the impact was not significant. And for the enzymes, in principle, we would expect higher enzyme activities under miscanthus. And for the nitrogen, we would expect to see the carbon cycling enzymes activity going up with as we increase the nitrogen. In contrast, we would expect a decline in the enzyme activities of nitrogen cycling enzymes as we increase the nitrogen. This is again part of the what are the nutrients required by microbes. In this case, maybe nitrogen is more in excess, so they don't want to produce a lot of these nitrogen cycling enzymes. We don't know or fully understand of that. I'm not showing a lot of results for the enzymes, but so far all of the enzymes I have measured, whether for carbon cycling or nitrogen cyclings, we did not find any impact of either cropping systems and nitrogen on these enzyme activities. And the responses of enzymes to these impact, whether it's nitrogen or cropping system, were variable and inconsistent. I don't know why yet. I'm trying to work on trying to understand it better. I haven't finished the whole analysis yet. So in conclusion, cropping system and nitrogen had little effect on microbial biomass and potential enzyme activity, but the variable and inconsistent responses of enzyme activities suggest that maybe the nitrogen effects are complex or dependent on other factors, maybe the duration of the study site, other factors related to the soil properties, microbial community, and how nitrogen impacts those factors. And also highlights the need of maybe we need to study these 2 things more and understand how they respond to nitrogen. And with that, I would like to acknowledge these folks, and I'm happy to take any questions.
Okay, let's start with questions for Alice, and she is. Well, first, why don't we go to our online friends? Those of you who are on Zoom, does anyone in Zoom land have a question?
We've got some chats. I don't know if they're.
Those are all from. Okay, anyone in the room? Question
This was very interesting, and it's kind of challenging some long-held beliefs and really environmental environments. wanted to ask about two of the graphs that show really high. One is the Trussell graph. The rates seem very,
Sure. Yeah.
seem like 1,000. Do they get 1,000?
Yes.
So, what are your thoughts on that?
Mhm.
And then the second one, but just the graph, just highlighted it. Here's your thoughts. Not on the ground source.
Sure. Yeah. For the first one, which is the Tricidar. Oh, yeah. So the question was about this figure by Tricidar 2008, that the nitrogen load was like insanely high. The other one about the mixed figure, and it was about the source of nitrogen and its impact, right? Did I get, okay, perfect. For the Tricidar 2008, I think I failed to mention that this is the cumulative nitrogen applied throughout the experiment. So yeah, that's really high. But that was the amount that was applied throughout the study period. And for this one, I wish I have a better answer, but I am aware of the source of nitrogen and how it impacts either microbes and enzymes differently. There's no clear answer yet, but I know that some, like if you're, for example, looking at ammonium versus urea, you might expect to see some different responses. And that's why maybe we should look at these more. I know we're still not fully understanding the impact, but yeah, I've seen both. Yeah, you're welcome.
Question.
Your lack of results make you wonder about the role of enzymes in plant production with soil plant system, or really can?
Well, I don't think I will panic because I see this as more like a challenging to think more about what do we see here. I know some folks like to have some clear, distinct results of what they're studying, but for me, if we focus more on understanding how enzymes function, the relation to microbes, and how they're actually collaborating with microbes when the nutrients are available or are not available, make us wonder how we can understand them better. So I think what I'm trying to say, the lack of significant responses so far may be encouraging to study them more, because I'm not showing the results here, but if you remember from this first study, we had higher nitrogen cycling enzymes in the prairie treatment with no nitrogen. For example, here I had one nitrogen cycling enzyme that was way high up at the very high nitrogen rate. So that leaves me wondering, like, why would this happen? Why I had the exact opposite pattern that I just saw in the other study. So I'm still wrapping my head about it. Hopefully I can get a better answer, what I should, before my defense. So I think, yeah, but there are still other things I have to look into it. I am planning to look at the oxidative enzymes, some more amino compounds, understand the availability of these amino sugars and amino compounds and how they relate to the enzymes or the patterns I saw. Maybe looking at the particulate organic matter and trying to understand how they all are related. And one more thing, I've read studies in which they reported an effect of nitrogen on these enzymes or microbes after 20 years of establishment. So with this being 7 years of establishment, I'm not sure, is it the duration? Is it the microbial community? Is it the negative or positive effect of nitrogen on these soil properties? So I don't know. I hope this answers your question.
Very good.
Okay.
Okay, thank you very much, Alan. We'll go on to questions from Matt's. Matt's, do you know? You project your voice really well, so I don't really think you need a microphone.
Well,
But,
for online.
Oh,
Yeah.
well, I bet can the online people hear me? I bet they can. Anybody saying yes, Laura? Okay.
Okay.
All right. So questions for Max. Anybody have questions for it? Okay, we got 2, 1.
But I didn't understand how do you use the camera and stuff. How do you prepare the sample impact cores or how do you?
Yeah.
Stand up and repeat the
Oh,
question.
yeah. So what the question was just how do you prepare the NIR samples for analysis? And so that's a lot of what we're currently teasing out, but what we're planning on doing is taking intact cores, breaking them up into small pieces, washing the soil off so you just have roots removed. From there, once we've got that root sample, there's a lot of variability in what you can do and what you're interested in. We're going to kind of start off by just grinding to a fine powder, our main body of samples, and using kind of, state-of-the-art lab-based NIR things. There's scientists working on NIR soil analysis in the field. There's a lot of very portable instruments. There's little tiny ones now that you plug into a computer and get, instant NIR data. But again, that needs calibration, and so we're kind of, like you said, grinding, making sure the sample is as pure as possible, calibrating on really nice machines before we move on it.
So by now you're going to wash the sample, keep the roots, and keep right.
Yeah, it's kind of got a screen that you put it on and shoots light through in the lab basically.
Other question?
It was the same.
Okay.
So you mentioned the need for calibration. How precise must your samples be to enable that calibration to apply to your samples? Is it just for the species, or is it the cultivar within?
So we are using the same cultivars, and I have not been able to find anything in the literature. I can give you my email if you have any good articles on kind of examining the differences within cultivars. But yeah, so we're calibrating to the specific cultivars that we have, both with the corn and the grasses. So hopefully that there's not crazy variety within that. But even if there is, it's still, I think, going to be useful information. And from everything I've read in the literature, we should be able to identify, even if the bands are relevant.
Okay, thank you very much. Let's go to questions for Jessica Nelson, who talked about water holding capacity. So,
Jessica, I'm just going to hear under measuring water retention active cores compared to destroying the structure.
No.
Under has a paper on the Bible share.
Yes, it is.
But he found that water holding capacity of the sperm soils was larger than intact structure. So I was a little bit curious why you had the hypothesis opposite of that. So,
That was for intact core versus aggregates. Our samples weren't as an intact core. It was just the soil that had been pushed through a 2 to 8 millimeter aggregates. So that is one reason that we thought, that's curious that I guess when the soil cores were destroyed, maybe the surface area is. I'm not sure what his conclusion were.
no, it's all about the pores.
It's all about the
So
pores.
if he destroyed the structure, the intact lost the large pores,
Right.
so that they didn't drain on the
Right.
disturbed sample.
Right.
They did drain, the large pores were there.
Right.
That's why I'm a little bit curious about how
Yeah.
you came to your hypothesis.
Yeah.
Seems counterintuitive.
Right. This is for me. It was a previous study that I was working off of, and we we're trying to look at I think that somebody had noticed that there was an increase, and then we were curious about why that was. So then we were starting to think maybe it's the pore space because they're both the same. They're both as composite samples, and that maybe it's the structure in the aggregates. Maybe those smaller pores are holding on to water, and when you destroy it, it's more uniform, and maybe there's less of that. So it's definitely a lot of questions, and I would think it would be interesting to look at more sites to see if the trend holds true and dive a little bit deeper into maybe some of them.
I'll say one more thing. So this thing is a question that I asked you last week. Was about the bulk density.
Mhm.
That's me. How's putting those on the very short sample? There's a very small range of the matrix. Right. The bulk density would affect that total porosity. To me, that would be like a really important component on how much water was left behind.
Right.
How they pack to different, pour them in the funnel.
If there
With
was
the
some kind
different
of
bulk
area.
densities, that's preparing something different. It's more on the packing.
Right, of the how close the particles are together versus.
Yeah.
Yes, that would be really interesting to look at. And do you think, how significant do you think the differences would be? Say you have the funnel, you have the sample, and you just dump it in. How daring could that really
You're
be?
gonna have to tell me.
Yeah, yeah, I'm just curious. I'm not sure how. I try to replicate my shaping motion, but
This is my advice.
Yeah.
I had last week too, is
right,
go back and look at it and see.
because instead of inside the aggregate, it's just the packing. Focus in the pack. Okay, that would be great. So thank you all.
I've got a question online from Dr. Cruz for Jessica. Would you expect similar results if you were addressing plant available water holding capacity instead of water holding capacity? So adding the plant available water. So
I'm not sure how that what we're really looking at with this type of water holding capacity is more of a container water holding capacity. So the water table essentially at the bottom of that container versus how it would be in the field with plants available. So I'm not sure how that would affect it because how we're looking at in the lab is we're removing it from the field, processing it, changing things. And so how that would affect the plant available water, I think would have to do with the pore sizes that we are looking at. And so when we're looking inside aggregates, those pores are really small. Those micropores are really small, which is not really that accessible to plants versus some of the larger pores. So unless we were measuring the size of the pore size distribution in our samples, I don't really know how we could tie that to plant.
Thank you, Jessica. Okay, our first speaker was Reid Barker. Questions for you? Phosphorous stratification.
Great, thanks for the presentation. I didn't quite understand one of those slides of the ridge soil surface concentration. On the flat soil, so they were around 60. I didn't quite understand why they were versus.
Sure.
Repeat the question.
So Dr. Horton's question was regarding the figure that I showed from Robbins and Boston on earlier slides was why was there a much higher concentration of surface to the more typical tillage system and no-till system? So the reason for that would be, like I mentioned, the lack of mixing. So broadcasted fertilizers, organic forms of mineralizable organic matter and manure, but over time they will mineralize. And since phosphorus is a very sticky element, doesn't very much in the soil, then it'll kind of remain in place. And then that's where the second part comes in. It's like when you don't till it, you're not redistributing that phosphorus deeper into the soil profile.
So it's a ridge. Thank you.
Other questions? For Reid?
Do the pea fertilizers, main ones, do they differ at all in their solubility and ability to soak down into the profile? I know there are people working on nanotechnology fertilizers that would be much more soluble for no choice. So I was wondering about the mainstream pea fertilizers, if they differ at all. How well we need them. Or are they all pretty much soluble?
So the question was, is there like the type of phosphorus fertilizer? Mentioned there was some fertilizer technology phosphorus, but some go deeper into the soil profile. As far as my understanding is concerned, generally it doesn't move. It probably moves. I'm not an expert on that subject, but I can give. Is generally it will move a little bit and then it will be quickly sorbed to either organic exchange sites and mineralized or mineral exchange sites. So it would theoretically be available and potentially see the profile, but over time, but very quickly grabbed by various soil factors.
The main pea fertilizers, they don't, there's different types of them, they don't differ that much. Like they're all subject.
It's
rapid source.
As far as
Yeah, I don't believe it. Okay.
So, is there, you mentioned that stratification is happening under the system. But for the same amount of phosphorus in the soil profile, is there evidence those stratified systems that plants are more peak?
So unfortunately, okay, so the question was, is there a lot of evidence to suggest that plant stratification or phosphorus stratification causing phosphorus deficiencies in crops? So unfortunately, there isn't a ton of evidence of that. It's there is some evidence. It's like one of the as far as I found, but that's actually one of the things in the analysis is generally what I found is that no effect or slightly negative effect on plant concentration of phosphorus. Again, in my thesis, I'm actually going to be comparing phosphorus stratification, what I'm measuring as ratios, and comparing it to plant phosphorus.
Okay, great. Thank you very much, Reid. And I want to thank each of you who has attended today for your participation in the seminar, and thank you all online too for coming. Thanks to each of the students. Really appreciate your talks today. We'll convene again in 1 week.